Hello, my name is Connor, and our group is going to be presenting on individualized DNA sequencing and the ethical issues associated with it. The agenda for today is to talk about a little bit of background about the technology, how it's being used in current day, the precedents associated with it, the regulations associated with it, and finally the ethical issues. So a little bit of background on how DNA sequencing as a whole works. First, we're going to differentiate between phenotypes and genotypes. So genotypes and phenotypes are essentially the same thing, except phenotypes are the observable characteristics due to genotypes. Genotypes are the genetic composition and sometimes are represented in a phenotype and sometimes they are not. And so whenever you're looking at something like 23andMe, they are presenting both genotypes and phenotypes. So some of the characteristics that they inform you about are in relation to your phenotype. So for example, like widow's peak, and in other cases, it has to do with something that is less obvious and is only seen in your genome. So to talk about some of the types of sequencing, to start off, there's whole genome sequencing, which is generally from scratch. So in this situation, you've got a new organism and it's being sequenced from start to finish, and you're using that genome in order to do some sort of research or just for uh, future knowledge. Resequencing is using a whole genome to basically compare to. So if you can sequence part of an organism's genome and then compare it to the whole genome that was sequenced prior to gain insight about the specific organism that you are studying. And then there's single nucleotide polymorphism targeting and exome pull down. Both of these are more targeted approaches where you are sequencing a much smaller portion of the genome and you're getting more limited information, uh, but it can still give insight into different traits and disorders and other genetic information. So SNP, or single nucleotide polymorphism, is focusing on, uh, as it states, single nucleotide mutations, I guess, in the target person's genome. And then exome pull down is targeting the sequences of protein coding DNA regions. Up here on the right, we have a schematic kind of illustrating how most DNA sequencing companies go about processing your DNA. So they use what is called a microarray, which is basically an assortment of traits that they are targeting for, and it screens all of those traits all at once. So that will give back feedback on both phenotypes and genotypes, depending on what they have actually put onto that microarray. Now, sequencing in the past, it's been around for a while, but the methods for which it's done have changed dramatically. Originally, back when the Human Genome Project was undertaken and completed in 2004, it took 13 years and around $3 billion. It involved over 20 different institutions, and overall the methods were slow and they took a very long time, which is a large reason why the cost was so high. Comparing that to modern day sequencing, you've got several different methods for high input sequencing. The most common one is called sequencing by synthesis, which essentially uses fluorescent tagging to measure each base as it goes along, and it does thousands of bases at one time. So then this process can happen much quicker. And since it's all done within a computer, the computation and measurement is much quicker. And so you end up with both a lower cost and a shorter time period. So approximately, to sequence an entire genome, depending on its size, takes about 2 days and around $3,000 to $5,000. And as of 2017, more than 500,000 human genomes have been sequenced. And the number is far, far larger now, especially if you include genomes outside of human genomes. And then up here on the right, we have a schematic giving a little bit of a preview into how traditional sequencing work, kind of like Sanger sequencing. And then down here on the bottom right, we have a graphic illustrating how the cost has changed over time. So as you can see, compared to back when the Human Genome Project was first completed, compared to now, that cost has dropped significantly. And lastly, since I don't have the time to go through and explain exactly how sequencing works, we've got a YouTube video that explains both how Sanger sequencing works and how next generation sequencing works. So if you're interested and want more information on the nitty gritty and some of the other processes other than sequencing by synthesis, that link is there for you. Next, Jamal is going to be covering the uses in biotech and in the technology.
Thank you, Connor. For this section, we just wanted to highlight some current uses of this technology. And firstly, we have personalized medicine, which makes use of individuals' medical information to provide a streamlined and effective treatment paradigm. Compared to current medicine, it analyzes all patient medical data available and tailors the treatment based on that information. This leads to a more positive effect seen in all individuals who undertake this method of treatment. Some technical challenges which involve personalized genomic medicine include the large amount of data which is being produced and the difficulties which come about in analyzing this data. The considerable amount of raw data which is being produced contains multiple sources of sequencing errors, such as errors in determining the nucleotide bases from the signal reader and mistakes resulting from detecting genetic variations in the sequence genome. These problems come about due to the fact that genome sequencing technologies are still in active development. Post-Human Genome Project research also demonstrated the complexity of human and other species genomes. The development of more advanced biotechnologies, more powerful computation, and more sophisticated data integration would lead researchers to discover many other previously unexpected factors involved in the development of disease. This will aid in the association between genetic variations and patients' risk of developing certain diseases, but it would also make it more challenging to analyze personal genomic data for healthcare purposes. Consumer genetic platforms have grown substantially over the past 6 years, with companies like 23andMe, Ancestry, and Nebula Genomics all making strides in this market. These services are straight to the consumer, with individuals seeking information about their ancestry, future risk of disease, or them being simply interested in helping genomic research. 23andMe and Ancestry make use of single nucleotide polymorphisms, which occur when a single base in the DNA differs from the usual base at that position. They employ genotyping, which compares an individual's genome with a reference one. This is only a snapshot of the genome being sequenced, while Nebula is providing a whole genome sequencing product, using cost and privacy as their top selling points. Although these services provide information relating to diseases and dispositions to disease, they do not diagnose any health conditions or provide medical advice. Genomic information has also aided clinicians in deciding treatment strategies, sparing patients costly and complex procedures based on a molecular diagnosis. Identifying how a patient will also respond to drugs has also been aided by genetic information, with researchers using genomic data to suggest new therapeutic applications for existing drugs and to better select individuals for clinical trials to find uses for drugs that failed earlier trials involving broader cohorts. Clinicians worldwide are also embracing far more comprehensive genome sequencing to search for variants implicated in undiagnosed genetic diseases and using this information to guide treatment. And relating to what I mentioned about the direct consumer genetic tests, although these tests are not accredited for clinical diagnosis, individuals are gravitating towards the use of these for personal genomic information. Lastly, we see the use of these technologies in criminal identification and record keeping with the use of APHIS and CODIS, which Maggie will discuss in the next section.
Thank you, Jamal. Now I'm going to talk about precedents that were set in the past. So in other words, how has genetic information been used and dealt with previously? So Jamal mentioned APHIS, and I'll talk about it more in depth. APHIS is the Integrated Automated Fingerprint System. This system is used by law enforcement and contains latent prints from crime scenes as well as voluntary prints that are taken from those who are arrested. But it's more than just fingerprint data. So it also contains mugshots, information about criminals, and information about military and federal employees, and pretty much anyone that Congress has interest in. This information can help provide positive ID of a person of interest, and that can include criminals or victims of crime. And this information can be accessed by law enforcement agencies and pretty much anyone who works for the federal government. Then there's also CODIS. So this is the Combined DNA Index System. It contains DNA from convicted criminals, people who were arrested, and DNA collected at crime scenes. So that can be someone who has committed the crime or anyone who has been at the crime scene in the recent past. It allows law enforcement to compare DNA samples from crime scene to crime scene and to individuals' DNA that is stored in the system. The system is accessible by law enforcement, and once an individual's genetic information is in the system, it's there permanently. So even if a person is found not guilty, their information remains in the system except under certain circumstances. And then finally, the last example I'll talk about of biological property being used is in human-derived cell lines. So for example, there are certain cases in the 1950s when cells were taken from patients to create cell lines for use in research. And this was before there was a requirement for getting consent to do this. These cells went on to be a part of many groundbreaking studies, such as the discovery of the polio vaccine, and the owners of the cells didn't have any idea that their cells were being used. But the patients and their families eventually found out about the use of the cells, and they filed lawsuits for ownership of their cells and for compensation of products that had patents that involved those cells. And then in the end, the courts denied them of ownership, and they did not receive any compensation. So these are just a couple of examples of how individuals' biological property is used and seen in the eyes of the government. With that being said, I'm now going to get into specific regulations that are active today. The first is the Genetic Information Non-Discrimination Act, or GINA. This makes it illegal for health insurers or employers from requesting or requiring genetic information. And then particularly in North Carolina, there is a law that supports GINA, but is more specific in stating that a health insurance organization cannot raise premiums or contribution rates or deny coverage based off of genetic information. So these laws are used to protect citizens from not having access to health insurance or an employment opportunity based off of their genetic information. There are some concerns for lack of regulation though. So GINA covers health insurance, but it does not cover life insurance. That can be dangerous for people who have genetics that insinuate they're at risk for death at a young age. And this aspect varies from state to state in the US, but right now Vermont is the only state that includes life insurance. In addition, there is the threat of surreptitious testing. So this means testing someone for their genetic information without their knowledge. And there are currently no federal laws prohibiting surreptitious testing. And with that being said, I'm going to pass it off to Jamal to talk about the ethical issues that we will be talking about in our synchronous meeting.
Thank you. So as Maggie said, we will be discussing a couple of industries that individualized DNA testing could be used and could lead to ethical implications. The first is healthcare. So receiving your genetic results and obtaining feedback from a healthcare professional. Then we have insurance, providing your genetic results to your insurance provider. Then there is employment/education, having this data be a requirement to obtain a job or to go to school. And finally, crime, having your information in a database accessible by law enforcement. Now, what we want you to do is think about which of these topics is of highest interest to you to talk more about in the synchronous meeting. We're going to have a comment for each topic below this video, and we want you to reply to the comment with your area of interest and start discussing the pros and cons for using individualized DNA sequencing in that industry. Just keep in mind that we only have 3 to 4 people per topic. So if 4 people have already replied to what you're most interested in, you would have to choose your second choice. Thank you for watching, and we look forward to this discussion on Wednesday.